Good morning, ladies, gentlemen, and participants. Good morning from the UAE. It is morning in the UAE. I would imagine it is afternoon in India, and in many other parts of the world, perhaps it is evening. Welcome to our 16th webinar. We have two outstanding speakers today. One is Professor Hafiz Ahmed from the RAC Medical Health Sciences University, and the other is Dr. Satyam Parmar, the head of pathology in our own RAC hospital. This is our 16th webinar, and our topic today is the role of the antibody test and the booster vaccine. Talking about the antibody test. Now, while the RT-PCR test and the rapid antigen test tell us whether we are COVID positive, or whether we have COVID, the antibody test tells us if after getting COVID or after being vaccinated against COVID, it tells us whether our immune system has produced enough antibodies to protect us against COVID. To talk about this in its entirety is Professor Hafiz Ahmed. And Professor Ahmed is very competently placed to talk about this subject. Dr. Hafiz did his PhD from the prestigious AIMS in India, India's leading medical university. He did his doctoral fellowship from the National Institute of Health in the United States and a postdoctoral training in molecular biology from the Abbott laboratories in Germany. Professor Ahmed is truly international. He has over 10 years of vast experience as an assistant professor of microbiology, clinical biology in the RAC hospital, in the RAC Medical Health Sciences University, and he is also the head of molecular microbiology at our very own RAC hospital. He has worked in New Delhi, India. He has worked in Germany. And as I mentioned, he is truly international. He has been an active speaker in over 80 regional and international conferences. Ladies and gentlemen, in order to unravel our understanding of antibody testing, I present to you Dr. Hafiz Ahmed.
Thank you so much, Dr. Andrea, and assalamu alaikum. Good morning, good evening to my colleagues and friends in all parts of the world. And thank you so much. It's really overwhelming to for such a grand introduction. I don't know, I deserve that introduction, not, but thank you so much. So without much ado, I'm honored to present this for the, as a mass seminar, especially because a lot of doubts are there in the community regarding with respect to antibody, the role of antibodies, and my colleague, Dr. Satyam, would be taking care of the booster vaccine and when and how to time those vaccines. So I apologize upfront if I overshoot over time, because this matter is still confusing, even though we are in the epidemic for almost 1.5, 2 years, the pandemic, but still we need, there's a lot of clarity required. That being said, I will just go ahead with the presentation, and if there's something you don't hear, if you technically you can't see, please, stop me over.
Doc, can I request to switch on your camera? Your video is not there.
It's switch on. Can you not see me?
No, Doc.
I can see you, Dr. Hafiz. I can see you from where I am.
Can you see the disclaimer?
Yeah, perfect.
Yeah?
Sure.
You can see the disclaimer now, right?
Right.
So at the outset, I'll be discussing a little technical as well and trying to just keep it a little simple, but I have no affiliations with any company or any sort of manufacturing antibody or COVID PCR corporates. And it's purely scientific what we discussed today. So we know the virus SARS-CoV-2, the name is SARS-CoV-2, which has evolved from nCoV-19 SARS-CoV-2, which was earlier in 2019. And we know the disease, it's COVID-19 is the disease, the virus being SARS-CoV-2. It's an enveloped virus, easy to kill because enveloped viruses are, just by soap and water, we can easily kill the virus. And that's what we teach students. Disinfection is easily done with an enveloped virus rather than a naked virus. We also know the host receptor, the ACE2 receptor, which is the binding site of the from the host side, spike proteins, which is causing a lot of interest because of the attachment between the spike and the ACE2. And since this is an RNA virus, it's always been a nuisance. Most of the viruses, especially HIV, influenza, dengue, these viruses are very difficult to to basically detect sometimes, also with relatively unstable genomes. And that's why vaccine efficacies and, it's difficult to get a vaccine, and like influenza, you may have to, top up the vaccines every year. So that being said, just a brief introduction. I'm sure many of you would know more than me with respect to the vaccine, with respect to the virus. We are more interested in what's called, I'll discuss more on the protein side, especially the spike protein and the nucleocapsid protein. With respect to the, when it comes to immunity, our body produces, when the virus enters the body, it evokes an immune response, and these proteins are a result of the immune response. But the timing of the immune response is different, and that's why it's very important to understand these proteins in detail and how we can, use this knowledge to especially understand the vaccine efficacy. So these are the major four structural proteins: spike, envelope, membrane, nucleocapsid. Sometimes we'll be using short terms, so you at least have an idea of NCP is the nucleocapsid, and is the most abundant protein produced. But this protein doesn't tell you about the efficacy of the vaccine, but tells you more about the exposure. And that's very important as we go down the lecture. You would come to get more idea on this. So these proteins basically they bind. Just to give you up head is that we have I will be restricting myself to the vaccine domain and the antibody testing. Thanks to the administrators here, we have almost 80% cover, 86% coverage of vaccine. And worldwide, as if you compare, it's pretty much high level. And that's why we have to understand the immune protection and and are we really protected, or are we just the numbers? So if you see the virus epidemiology, or the virus, the timing of these tests, in the incubation period of the virus, where the first one, the most sensitive test is the PCR, the real-time PCR, which comes just before the onset of symptoms, and then it peaks down slowly. Then you have this virus culture, which is very difficult to do unless you have a biosafety level 3+ laboratory, and then what we have is these antigen tests. So antigen tests are also employed in the hospital. Our hospital we do, especially for emergency cases or electives. And the last, which comes in after 2 weeks or so, is the antibodies. And we are mostly today interested in antibodies testing and how the dynamics of antibodies. So coming down to the antibody section, we have this nucleocapsid, the spike, which consists of spike 1 and spike 2. It's a trimeric region which also has this receptor binding domain. And then we are looking into antibodies, either total antibodies, IgG, IgM, IgA, but mostly relevant one is the IgG. So far, the data, corroborates with more with IgG rather than IgA or IgM or total. And we have we just don't want to know the spike antibodies produced by the body. Also, we would like to know the functional, those antibodies of the spike, which have functional ability to inhibit the virus. And that's why we do these, neutralizing assays. That's the whole purpose of doing neutralizing assays as a reflex of spike testing. One of them would be a plaque neutralization test with a live virus, which is very dangerous, and only few labs in the country would are capable to do that. Then we evolved with pseudovirus tests. We evolved with different artificial virus tests, and of course now we have ELISA, the enzyme-linked immunosorbent assay with recombinant receptors and recombinant proteins, which has given us the way forward for understanding this whole, neutralizing ability. So this is the structure again. The spike consists of spike 1 and spike 2 and the receptor binding domain. The spike 1 and the receptor binding domain are the main ones which are evoking the immune response or which are attaching with the ACE2 receptors. And that's why as a laboratory or as a functional, we want to know the immune response based on these two parts of the protein. So that being said, I'll go ahead with the technology. There's so many tests in the market, and it's very important to know which test have a high sensitivity. Nowadays, even ICMR India as well as here, the lateral flow assays are being very rampantly used, but they're not very sensitive, and that's why we have to go with the higher technology, especially ELISA or the chemiluminescent enzyme immunoassays, which are the mainstay in the diagnosis. And these are more or less quantitative, tests which can tell you the number of spike proteins and also the quantity of the antibodies, and especially the neutralizing antibodies. Because all these tests are not equal. Each test we are looking into the domain of more of personalized care, especially when it comes to COVID, antibody testing, because everyone has a different immune response, a different innate immune response, and he or she, behaves differently to the virus. The same brother and sister may get one brother may get a severe infection, the other sibling may just pass, just like a flu. He may pass because of the innate immune system also has a big role to play in this. That's why we need to understand the epidemiology. Also, which protein are you testing? Are you looking at the nucleocapsid? Because that's only going to tell you about the epidemiology or the exposure level. But if you're looking at spike is the one which gives you the idea of the surrogate protection, which especially the receptor binding domain and the S1 are the key molecules we look for. I'll go through this because there are a lot of slides I get to cover, and that's the key for us to hear for today. And in the vaccine era, post-vaccine era, is spike antibodies and measuring the spike levels. Also, the measuring the antibody or neutralizing antibody to the spike levels, because you want to know the overall protection, at least one arm of the protection, the humoral response. So what happens normally is in a normal situation, the ACE2 receptor binds with the binds with the virus, and you have the infection going on. But in the case of when you have neutralizing antibodies in your as a result of your infection, a natural infection, or as a result of vaccine, then these antibodies will bind to the spike, bind to the spike and prevent the binding of the ACE2 receptors. So it will just overall encircle the virus and does not allow it to bind to the human ACE2 receptors, thereby preventing the infection and give you protection. And that's the whole concept of neutralizing antibodies. I hope I would be loving to take questions, but the whole idea is to if you're not just your spike antibodies being produced in the body, but how much of ability of those spike antibodies have the neutralizing ability. And that determines your functional antibodies. That's the neutralizing antibodies. So it's a test which is required, especially in the immunocompromised population. And there are many different ways we have evolved with the tests. Now we have pseudovirus or neutralization assays. A lot of laboratories are doing ELISAs with the ACE2 receptors, and the different companies, GenScript and Euroimmun and many others have come up with these tests. I'll not go into the details, but we're not doing a plaque neutralization assay which measures the pox. You need a live virus because every lab is not capable of doing it. It's it requires BSL-3 or literally NASA level kind of facilities for a very high biohazard level to live culture the virus. So therefore we rely on pseudoviruses, viruses which are recombinant, and we do these tests in the laboratory. RAC Hospital is one of the laboratories. Now in the Nether Emirates, we have evolved these tests, and we have been doing almost more than 200 to almost 2,000 tests so far, and which can give you the level of your neutralizing antibodies and how protection you have. There is a validation, and there is, make sure whenever you choose a laboratory, you have to idea, is the test validated, and how good is the technology, because there you have to quantify the neutralizing antibodies by these tests, and very few labs are capable of this. And there is literature available, especially, but it takes a little bit time initially. Nowadays we're doing with ELISA, and this is the best test, but the best way to know the antibodies levels. So I don't want to go too much into detail of the evolution of these antibodies, because most of you mostly IgG, IgM, almost they come at the same time. But we need to understand the neutralizing antibody levels, and that's why, again, when I come back to the graph, that as a clinician, a clinical decision maker, you need to understand the dynamics of the PCR, the PCR, and the limit of detection of these assays, because the timings may be different for different tests. And that's where, if you're looking at antibodies, you have to look at least 1 or 2 weeks post-infection. So there are CAR tests available, but they are not so sensitive. And this is the principle of ELISA. We teach our students antigen complemented by primary and secondary antibodies, giving a substrate. And there are many tests in the market, but we are more interested in the ELISA and CLIA platforms, which also gives you a spike as well as neutralizing antibodies. And there are many companies, without any bias, I would say, but we have to look into the concept that, is it nucleocapsid which gives you the exposure, or is it the spike which gives you the protective antibodies? And especially looking at the, because this is a major concern. Some people say that some labs are doing N protein or nucleocapsid. Why aren't you doing it? It's because that you don't need, you are vaccinated. You don't want to know your status, what happened 1 year, 2 years before. You want to know your current status, and that's why you want to know your spike antibody or levels, or neutralizing antibody levels. And this is basically, the whole cycle where the antibodies develop, especially 1 to 2 weeks after the infection. And most of these, sometimes they are also age dependent, especially in people who are elderly. It is advised to do these tests because, as we know, the titers of these antibodies go on decreasing. So it is a multifaceted approach we go with, because there's something called as binding and non-binding antibodies. Just a little bit more technically, we are looking at neutralizing antibodies, especially which are leading to the loss of infectivity and blocking the virus from entering into your body. And that's where many commercial assays are not able to distinguish it. So it has to be validated, and it has to be it requires some expertise to do these tests, especially. We also do antigen testing at RAC, and we have an experience of, but we find that they perform better with the same antigen and same material VTM was used. Especially the chances of false negatives are higher if you use a different one. UAE has come up with a good guidelines, or if you're interested in looking at these guidelines, and you can have a look at, and what they've given is, what I'm trying to say is, if you are negative, if you have a negative, if you've taken the vaccine and you still are not producing antibodies, then do have a consultation and do a test, especially you do a neutralizing test with the spike 1 and the receptor binding domain to know if you are not producing antibodies, especially the timing of antibodies. If you're not producing the antibodies within 14 days of the vaccine or of exposure, then the prognosis is not good. It's a poor prognosis, especially. So the anti-spike estimation post-vaccine gives a better correlation, especially the S1 and S2. And there are especially some kits also coming with the trimeric proteins. This is a slide which is more of vaccine related, and the Israeli study quickly, in summary, summarizes that if you're vaccinated, you have less chances of, your viral loads will be less, even though you're infected, your chances of, getting a severe infection is very low, especially in this whole. I think Dr. Satyam will be speaking more on this. I will be concentrating a little different today, especially why are we doing these tests and what is the need today? Because we know a lot of variants are coming up, and there's something called as deletion mutant, especially if the variant which you are looking, Delta, or a new variant which emerges, if the spike region has a mutation, especially in these 69, 70 amino acids, there is a chance that the some of the PCR kits will give you false negatives. And MOH has given already an advisory saying that don't use a kit which has single gene, because you may be missing, because your spike gene may be mutated. And that's where also it relates to your antibodies production, especially if the mutation is there in the receptor binding domain. What happens if they see the spike protein, I think the peptide is a spike, especially if these mutations are this particular codon, it changes the conformation of the protein, and basically the virus basically behaves differently, and the neutralizing antibodies are more resistant, and that's the key to the whole assay. So we are looking at protective immunity, and if you're vaccinated, you need to if you're a frontier worker, if you're immunocompromised, if you're a cancer patient, an HIV patient, it is advisable that you do these tests, especially to know your protective immunity, and especially in times when you have emerging variants. The variants may vary differently. As also evident is a paper, a very nice research which mentions that if you are elderly and the immune response is age dependent, especially they did with a study with Pfizer, and they found that a booster is required, especially in people who are 65 and plus, because you have neutralizing antibodies which are less produced in these populations because of the immune response. And there is a plethora of evidence. Science supports the theory of antibodies, although it's just a single component, and there's also a cell-mediated component to your immune response, but this is a surrogate marker, and you get an idea, especially in elderly population, if because there's a rapid decrease in immune response, and therefore a booster may be required to keep up the levels. I think this will be discussed by my colleague, Dr. Satyam, whether mixing and matching and requirements. I will be concentrating a little more on the variants and why alpha, beta, gamma, delta are so important to us, especially in times of vaccines, because many of them behave differently. And there's something called as vaccine escape mutants. And this is a study where I come from AIMS. The variants of concern, basically, they might break through. Even though you're vaccinated, you've taken your double doses, still there is a chance that there may be escape mutant, and you may get reinfection, or it can be a repeated infection with the same virus. So there's a big good study which says that the viral loads are very high, especially with respect to if your Delta variant versus a normal Wuhan virus or other variants. And that's why the whole disease epidemiology has changed. The number of deaths has been very high because people were not vaccinated, and the Delta called the havoc. So therefore, booster, it's not a uniform, or universal thing. But of course, if you're aged, elderly, and you may be having, the antibody-resistant variants, and therefore, or also if you're immunocompromised, these, boosters may be required. Some people say that I'm fine, I'm healthy, I have a very nice body, but if you see the studies, they say that almost 74% transmissions occur asymptomatically, pre-symptomatic, and that's why which caused a lot of Delta havoc in the world. And the whole, if you see the vaccination in US, this is just some data, they say that if people who were vaccinated, the incidence of these infections was very low because of this high vaccination coverage. So we need to have the safeguard because of the variants, because the variants behave differently in terms of transmission, in terms of producing the antibodies or neutralizing antibodies, reduced neutralization, is seen in some of these variants, and that's why a booster, as the government and the WHO and the CDC are saying, is evident from the science. So variants of concern especially require high level of neutralizing antibodies. I googled some latest information I found very interesting, that the reduced antibody neutralization, if especially with respect to different variants, are seen, and therefore the concept of giving a booster vaccination to maintain high neutralizing antibody levels. So there's a type of evidence with respect to vaccine efficacy, and you can correlate with the protection. You can do the test and keep a safeguard of the vaccine breakthrough infections, because a single vaccine breakthrough infection can make a hotspot in that area, and that area can, it can spread. As you know, a single hotspot can cause whole country or maybe countries to have the infection. As now you see Europe and Russia and many other countries, they are still facing the same problem of 50,000 cases, 1 lakh cases a day. So to date, available demonstrable antibody, we know that the antibody declines with time. 8 months, 6, 9 months is the time period we have the data so far. And with respect to different vaccines, especially Pfizer, Janssen, they have some data after 6 months. But to maintain a long-term protection and waning immunity, there is a booster which is required to maintain the neutralizing antibody levels. A lot of data have and some cohort studies, especially which mentions from Israel the higher breakthrough infection rates, especially with some vaccines versus the other. That's why the concept of mix and match is coming, and that's why we need to attack the virus from different sides to have a better protection. And that's the whole idea for the vaccine efficacy and the breakthrough infections. Without taking still more time, we just need to understand that all the immunocompromised people and the unvaccinated people are more at risk of these emerging variants, and they have to be there needs to be a safeguard, especially with respect to the emerging infections. I will skip some slides just to show what exactly I have more coming up. There is another angle to immune response, and that's the cell-mediated immunity. But we are measuring more of humoral immunity, the antibodies, but also your cell-mediated immunity tells you, even though your antibody may be less, but still you see many people are protected. And one of the reasons is the immunological response, or the subset of memory T cells and B cells, which remain more after 6 months of time in the normal population. But there are also some assays which are coming up in the market, and we also try to validate some of these assays. So we can see the picture in totality, the antibodies as well as the memory response. But there is a cross variation protection available with these antibodies, and these neutralizing antibodies can be boosted with 1 or 2 different vaccines. And there's a very nice study from Nature I saw, and I just wanted to share with you that they have did in macaw monkeys the viral challenge, and they found what they found was they found a cutoff. Because there's no answer to how much of antibodies should I have to be protected. There's no standard cutoff, but they say that any antibody levels, high antibody or medium or low, that's the only grading we can give. So anti-spike 400, anti-receptor binding domain 100, and neutralizing antibodies 50 plus is what they have given. Now we consider nowadays 35% of inhibition. If you have, then you are more or less protected. So so the whole idea is to protect the virus from binding to our cells by neutralizing, by functionally neutralizing it with developing a good immune response is the key for development of neutralizing assays. And it correlates very well. What is more important, very nice research has come up, the timing of these antibodies. If you're not producing these antibodies within 40 days of disease, then the prognosis is not good. So if you're taking a vaccine and if you're not producing the antibodies in this time period, then definitely you can consult someone a doctor or try to have your titers done. That's the whole idea of this vaccinology or these, because there is a timing of immune response. If your body is producing the neutralizing antibodies delayed, it means that the immune response is not so active against the virus. So why to get this? When to get it? After vaccination, almost 30% inhibition is good enough. You don't need to achieve 100% as but 30% is good. And if you don't have antibodies, it may indicate a poor prognosis. After infection, also monoclonal antibodies are based on these spike antibodies, and if the neutralizing antibodies are present, then some of the doctors they differ monoclonal antibodies. Or if someone is seronegative, then the monoclonal antibodies is given to them, and it reduces the hospitalization. And I think at RAC, with our leadership, sir, and the Zionist initiatives, we are doing a wonderful job also of giving monoclonal antibodies. By now, you know what neutralizing antibodies are. They can block the infection and they can neutralize the virus. And there's a lot of evidence. If you see this, the breakthrough infections with respect to the different vaccines, you would find that the booster or some of the vaccines, when given after a delayed vaccine, gives a long protection, and especially against the variants. And these are some of the studies you can go through, and they basically they tell us that because of waning immunity, a booster is required, and especially neutralizing antibodies offer greatest protection from reinfection, because if you are reinfected, sometimes the infection may be more persistent or more symptomatic, and therefore these studies which say that you are 2 to 3 times more risk of infection if you're not vaccinated. So vaccination is the key to keep up the population from being safe, and quantitative measurement of neutralizing antibodies. Why do you want to test it? Because many people, I just took this from a Siemens person who was delivering a presentation, and very nicely they mentioned that higher levels of antibody are more protective, and the data shows this. There's an evidence of showing this protection. Especially, they correlate very well with the binding assay and the protection immune response in the Oxford study, and there are many other studies, especially they have published. So, what are you looking for? Are you looking for IgG, IgA, or total? The data supports good evidence of IgG in totality is enough for measuring the protective immune response or spike antibody levels, and not this nucleocapsid protein. So this is the message from this, and IgG is the dominant, post vaccines, but the levels may vary from person to person, from vaccine to vaccine, or from the timing of your vaccines. So you can benefit from these tests. And I just saw a very nice, again, new paper, which from Nature, Lancet actually, which mentions that if you read through the paper, the immune response for booster, they also mentioned at the end of the paper that serological testing in future might be used to identify who has COVID-19 infections and prioritize boosters, which we are currently doing it. So we are doing it in the right way of prioritizing patients who need boosters by using these tests in the laboratory. This is just a copy of the report which we give. If you see, and a lot of queries come, how much protection do I have? How much results? If you're having more than 50 plus, you're positive. You have good spike antibodies, and you need not have the maximum. And the next one is the neutralizing antibodies, or the inhibition, how much you have. So inhibition of 35 plus is positive. If you're having less, then definitely you are a candidate for booster, and you may consult a doctor. So this is just one of the reports, how to interpret the report, and you're always welcome to visit us and see us, myself, Dr. Satam, anytime to have any queries regarding these. So CDC is monitoring these effectiveness data, and we have also data coming from UAE. Very soon to be published. These are some of the slides. What we need to be keeping safeguard on the emerging new variants and the reinfection monitoring, the contact tracing, because these, if we allow any one person who is non-vaccinated, he is the key for us to get vaccinated, and a person who is as immunocompromised is the key person for getting a booster. So, but this just some conclusion slides. We have done almost 1 million tests for COVID. also neutralizing antibodies, almost 1,000, 2,000 tests we've done. Some assays for the T cell and validation. We are also doing BAMLA, the first ones to do a BAMLA. We are also Regeneron treatment with a good, with a good team of physicians. Luckily, with our team, I wrote a proposal for, brain sequencing, and we have been granted the proposal for the first study in Ras Al Khaimah to do a genome sequencing for COVID variants. So the key message is detect. Still, we need to keep our guards for the genomic surveillance of these vaccine breakthrough infections, especially, and to understand the booster vaccination requirements in the community and the protection levels is a key. This is our team, wonderful team. We've been almost together since the epidemic, and each one of us has contributed equally. I'm just speaking here, but each one of this team is equal contribution. With this, I thank everyone, and thank you so much.
Thank you so much, Dr. Hafiz. Thank you so much. A lot of information in a little time. Thank you so much.
Thank you.
Now, our second speaker. Our topic is the booster vaccine. Now, many of us have had our COVID vaccine shots. 86% in the UAE have in fact had our COVID vaccine shots. And if you have not, my strong suggestion is that you should have your vaccine shots. But what about the booster shot? The question is, should we have it? Who should have the booster shots? What are the geopolitical implications of the booster vaccine? To answer all these vexing questions is none other than our Director of Laboratories, the Head of Pathology in RAC Hospital, Dr. Satyam A. Parmar. Now, Dr. Satyam completed his medical degree and his MD in pathology from Rajkot in India. He has over a decade of experience in hematology, clinical chemistry, immunology, clinical pathology, and there are many other words that I cannot pronounce. I think it is cytopathology and histopathology. That is a vast range of expertise. He has comprehensive knowledge of the laboratory and has developed his own basic laboratory information management solutions. He is also an accreditor in the ISO 15159 with JCI, TMOS, and others. He has authored various articles and published, which he has published in many international journals on the study of human tissue tumors, Kindler syndrome, and several other subjects. Ladies and gentlemen, I present to you our Director of Pathology, Dr. Satyam, and he will be advising us about the booster vaccine. Dr. Satyam.
Thank you, Professor Kennedy, and good morning all for having the wonderful meeting today. Dr. Hafiz has covered so many points here, so might be my points will be overlapping here. So what is my the thesis of my the presentation is that the main aim was presentation. So what is the basic vaccination and what is the requirement for the booster shots? So, I think my presentation is visible now.
Yes, Doc, we can see your slides very clearly.
Thank you. So the content of this presentation will be COVID-19, a small part of COVID-19. What is the vaccinations and what should you know about the vaccination and the booster shots? As we already know what is COVID-19. It is caused by a SARS-CoV-2 viral infection, and the range of illness will be mild symptoms to severe illness and death. The exact mode of transmission is not yet known, but what is the presumed part of mode of transmission is by sneezing or droplet infection, by the aerosol transmissions. Some people, such as adult about 65 years of age and older patients, and certain people with the medical conditions are more likely to get the infections easily than the normal people. How to prevent the COVID-19? Wear a mask that covers your mouth and nose. Avoid close contact with the others. Stay with 6 feet distance. Avoid crowded and poorly ventilated spaces. Wash your hands frequently with the soap and water, at least 20 seconds. Use an alcohol-based hand sanitizer, which is having the alcohol concentration more than 60%. Avoid frequent touching of your eyes, nose, mouth with the unwashed hands. Clean and disinfect frequently touched surfaces in your areas. And the last and not least is get their COVID-19 vaccinations. A recent statement released by the FDA commissioner 2 days ago about the COVID-19 vaccination. As the pandemic continues to impact the country, science has shown the vaccination continues to be the safest and most effective way to prevent COVID-19, including the most serious consequences of disease such as hospitalization and death. The available data suggests the waning immunity in some population who are fully vaccinated. The availability of these authorized boosters is important for continued protection against COVID-19 disease. It means that the vaccine are effective against the COVID-19 infection and the spread of infections. And as we know, that is the waning immunity, the immunity which we just achieved with the vaccinations or by natural infection. It will wane by the period of time. And that's why we need a booster shots in especially indicated populations who are most vulnerable to get the COVID-19 infection. The data in the UAE is showing that how many patients are already got this one dose and who are fully vaccinated, who has taken all 2 doses. You can see the total doses given here is 20.8 million people. The people fully vaccinated is 8.58 million, and percentage is 86.7% of the UAE population has already got the vaccination. So what is the vaccine and how does it work? The vaccine prevents the disease that can be dangerous or even deadly. Vaccine usually do not cure the disease, but that will help to prevent the disease development of our immune system. They will help to boost the immune system to produce the antibodies, just like a natural infection happens. So after the getting vaccinated, you have the protection from the disease without having get the disease first. So before understanding the antibodies produced by the vaccine, we also understand what is the difference between the antibodies produced by the natural infection of COVID-19 and what is the antibodies produced by the vaccine. So the infection induced immunity to COVID-19 by natural infection. It lasts many months and it is multifaceted, generate antibodies which are having protection against the nucleocapsid, matrix protein, and envelope protein. The natural infection produced the antibodies because the infection you are getting by the nasopharyngeal, means the upper respiratory tract infections. So you are having both the immunities like immunity, systemic immunity and mucosal immunity. So what is the vaccine-induced immunity to COVID-19? So vaccine-induced immunity to COVID-19 is the neutralizing antibodies against the spike protein. The SARS-CoV-2 is when the SARS-CoV-2 is encountered naturally in a vaccinated people. These neutralizing antibodies will bind to SARS-CoV-2 spike protein and block the virus from entering into the cell and multiplying in the cell. As we all know, the multiplication of the virus, they need some kind of medium, that is our human cells. So if we are not able to enter the human cells, they will not able to multiply by themselves. So the role of vaccination is to prevent the entry of the virus inside the cells. in a few people of population where there's a weaker immune system, like in the elderly people or those who are having underlying health conditions, they will have a delayed response or maybe a subnormal auto antibody response after the vaccinations. This is a basic difference between the mucosal immunity and the systemic immunity. Like when you are having the vaccination, we are giving is the intramuscular injection, which can go to the body and will produce the systemic immunity. The nasal COVID-19 vaccines are still under development, which can also help to protect for the local mucosal immunity. Neutralizing antibodies seem to correlate with the protection from symptomatic SARS-CoV-2 infection. So that means that if we are having a vaccination and if you develop a disease, the severity of the disease will be very much mild. The data suggests that the antibodies against the SARS-CoV-2 persist for at least 6 months after vaccination. After 6 months, there is a waning of neutralizing antibodies is reported, and however, it is unclear if the declining antibody titers are indicative of declining protection. At present, it is unknown that what level of neutralizing antibodies or other immune markers are associated with a vaccine's protections or infection or severe disease and transmissions. We can go through with some basic aspects of what is a vaccine, how it is being developed, how it is the efficacy. So worldwide, the scientists, the industries, and the other organizers work together to develop the safe and most effective vaccinations. The vaccinations are developed by understanding the genetic code of the SARS-CoV-2 virus, and based on that, the different kind of vaccination, for example, is the RNA vaccinations, the whole inactivated virus vaccinations, or by the inserting the viral code into the other viruses, which is known as viral vector vaccinations. So till now, more than 270 vaccinations has been developed, with over more than 50s being given to the people in the clinical trials, and more than 11 has been trialed in the large phase 3 trials. Some vaccinations has been made using currently used vaccine technology. Others have been made using the approaches or the methods used to, like a previous vaccine developed in the SARS pandemic and Ebola. So what is the basic property of the vaccinations? What is the requirement of the vaccinations to be introduced into the population? So the ideal vaccination will have the same immune response or the protection which is done by the natural infection without causing the disease. It will generate the long-lasting immunity so that the person is protected if they are exposed to the infection. It will interrupt the spread of infection by preventing the carrier of the organisms in the vaccinated people. These vaccinations to be safe, and the risk from the side effects should be much lower than the benefit of preventing the death or the complications of the disease. So that means that there is no vaccination without any side effects. There is some side effects, but we have to see what these side effects are beneficial compared to the complications or the other effects of the disease. And for the COVID-19 vaccinations, most of the vaccinations, these properties can be confirmed from the clinical trials. How we can know that the vaccines are safe? The vaccine has been given in a different trials, 3 trials. The first trials or the preclinical trials where the small group of the people receive a trial vaccines, followed by few hundreds of the people, which is given a specific group of the people to see the conditions of the vaccinations, effects of the vaccinations. This is followed by the phase 3 trial where the thousands of people has been given to trial vaccinations to understand the effectiveness and safety. All these data are presented to CDC. CDC is the Center for Disease and Control, USA based, which knows this data, and it will find out whether the vaccine is safe and will forward this data to US FDA. The FDA will look at this data and will advise from the advisory committee and decides whether to approve the vaccinations for the general public or not. The vaccination is only approved when all the 3 steps has been done and various teams of reviewers are sure that the vaccine is safe. Some commonly available vaccinations like Sinopharm, which is available first phase in the UAE, which first introduced with the whole virus inactivated vaccine with the 2 shots 21 days apart. AstraZeneca, which is a non-replicating viral vector vaccinations, 2 shots with 8 to 12 weeks apart. Pfizer vaccination, which is the mRNA vaccinations, 2 shots with 21 days apart. Moderna, again mRNA vaccinations with the 2 shots 28 days apart. And the Janssen, which is a viral vector vaccination with the only 1 shot. These vaccinations have reported the sustained effectiveness against the severe COVID-19 after 6 months. Means even after 6 months, if you have contracting a disease, you will have a very little side or the symptoms or the signs of the infection. While breakthrough infections are expected, the vast majority are less severe than those which are unvaccinated people. Okay. Will the vaccines are effective against the different SARS-CoV-2 variants? The answer is yes. It's normal for the virus to change as the spread. So what is this mutations? First, the mutation is when the virus is changing their antigenic property. The virus is not changing the antigenic property within a year or within a month. It will require so many years to completely change the viral structure, which is known as a genetic drift. So the vaccine which has been developed against the first mutation, which also affect against all the variants. So studies suggest that the vaccine provide protection from the non-variants up to the Delta variant. Even if vaccinated people get infected with the COVID-19, they are very protected against the disease. So these are some common differences between the Alpha, Beta, Gamma, and Delta variants and how the effectiveness of the COVID-19 vaccinations. So if you can see the last part, the COVID-19 effectiveness of the COVID-19 vaccinations is equal to all the variants in the Alpha, Beta, Gamma, and Delta. So coming to the main topic, why we need the booster shot. What is the booster shot and which are the eligible people which need a booster shot? So what is the difference between the booster shot and additional dose? A booster shot is administered when a person has completed their vaccine series and protection against the virus has decreased over the period of time. Means the patient has taken the 2 or 1 shots of the vaccinations, like 1 shot is only for the Janssen and remaining vaccinations is having the 2 shots. So totally completed the series and required, and it will show that the protection against the virus is decreased by detecting your neutralization antibody titer. The additional doses are administered to people who with moderately to severely compromised immune systems, and this additional doses of mRNA COVID-19 vaccine is intended to improve in the immunocompromised people's response to initial virus vaccine series. So we know the two terminology, waning immunity and poorly primary response to vaccinations. The waning immunity means the third dose should be prioritized for the vulnerable people, those who are at risk populations, like in the hospital setups, or is there evidence of the waning immunity against the severe disease and death. These people are not fit and healthy. The poor primary response to vaccinations is immunocompromised people who are not responding sufficiently to 2 doses of the COVID-19 vaccinations. For example, in the organ transplant recipients, which are having the immunity is lowered and they are not able to develop the proper immune response. Emerging data shows that immunocompromised people should receive a third dose if they did not respond sufficiently to the initial doses, or if they are no longer producing the antibodies. So who are these target people? The people above 65 years and older, residents of long-term care settings. Long-term care settings means people are admitted long-term in the ICUs or in the other healthcare facilities. People aged 18 to 64 years with underlying medical conditions, which is defined by the CDC, like cancer, dementia, the patient who are having the chronic lung, liver, or kidney diseases. People between 18 to 60 years who are increased risk of COVID-19 exposure and transmission because of their occupational and interstitial settings. You are still considered safe if you're fully vaccinated if you don't receive a booster dose. It means that it doesn't mean that if you have not received the booster dose, you are not safe. The side effects following the booster shots are similar to those which you are getting in first or second doses. You can get a COVID-19 vaccine and other vaccinations along with the flu and Shigella vaccination at the same time or close together. So there is no a period of the leg is required to have the two shots between the booster shots of the SARS-CoV-2 vaccinations and other vaccinations. Now, what is the concept of mix and match booster dose? Means, for example, you have taken a two series of one vaccinations like Sinopharm, and after that you are going for the booster source of the Pfizer's. Or if you have taken a Moderna vaccinations and you are going for a Pfizer vaccinations. So FDA very recently authorized the use of heterologous mix and match booster dose for currently available FDA approved vaccinations. A single booster dose of any of the available COVID-19 vaccine may be administered as a heterologous booster dose following the completion of the primary vaccination. Means you have to complete the first two series of your primary vaccinations, and then you are eligible to get the booster shot. For example, if you completed Sinopharm vaccinations 2 doses, if you want to go for the booster shots, you have to finish 2 shots, and after that minimum 6 months period is required to go for the booster shots of the Pfizer. Some of the frequently asked questions which can come during the time of vaccinations. Can I get the COVID-19 infections when I just getting the COVID-19 vaccinations? So the answer is no. The vaccination usually does not contain any live virus, so it does not create any COVID-19 infections. Does the COVID-19 vaccinations are having any side effects? Yes, but they are not serious side effects. It is possible that some people may have the side effects which are normal signs that your body is building the protections. These side effects are very mild and limited to tiredness, headache, pain at the site of injection, muscle and joint pain, chills, nausea, and vomiting along with the fever. Should someone get the vaccinations which having the allergies? You should first rule out what kind of allergies you are having and what is the content inside the vaccinations. So if you are having the allergies to the contents inside the vaccination, you should be consult with the medical doctor whether to go with the vaccinations or not. If you want to know which vaccination is containing which kind of content, you can go with the individual vaccinations sites. So should I get the booster shot even if I'm not listed in the recommended groups? For example, in older age patients, or if we're not immunocompromised, or if we are not working in the occupationally high-risk people. Yes, additional populations are still recommended to receive a booster shots as a more data become available. Experts are looking at all available data to understand how well the vaccines are working for different populations. If we need a booster shot, is working well, the vaccines are working well with this one. So the COVID-19 vaccines are working well to prevent the severe illness, hospitalization, and the death, even against the widely circulating the Delta variant. What are the risks of getting the reactions? So far, the reactions are reported after getting the Pfizer vaccinations were similar to that of the 2 booster shot of primary series. The fatigue and the pain at the site of infection is the most commonly noted allergic reactions after the booster shots, and everyone is still considered fully vaccinated after the 2 shots of their second dose in the 2 shot series. So what is the take-home message in this? So what to expect before, during, and after the COVID-19 vaccinations? Before, you just learn about what is the COVID-19 vaccine. See, COVID-19 vaccine is recommended, which kind of COVID-19 vaccine is recommended for you. During the vaccinations, read the fact sheet if you are having allergy to certain kind of allergen component inside the vaccinations, take your vaccination record card. After the vaccination, you are having some kind of side effect, which is very minimal, and continue using all the measures if you are even vaccinated. So it doesn't mean that you should not taking all the measures to protect yourself and the others. Protect yourself, your family, friends, co-workers, and your community, and get vaccinated. Thank you.
Dr. Satyam, the sign of a brilliant talk is a complicated subject simply delivered. And that was very well done. I understood every word you said, Doctor. Thank you so much. That was very well delivered. Now, I am going to take advantage of being the anchor, and therefore I am going to ask you a quick question before my colleague, Dr. Wilku, takes on the question answer session. And that is, Dr. Satyam, now I've had the Sinopharm several months back, and 6 months later, I took a single booster dose of the Pfizer vaccine. My question, Dr. Satyam, is, do I go in for a second dose of the Pfizer vaccine?
Ideally, this is not required. As I just told, the booster shots is required in a specific age group peoples or the people who are having the vaccinated people. As these are having immunocompromised status. So even if you just understand the basic role of the vaccination is that when you are getting the vaccinations, your memory cells are becoming sensitized to the antigens. So the whatever the antibodies are produced, whether it is a neutralizing antibodies or the spike protein antibodies, they are having a definite lifespan. For example, they are about 72 days to 75 days. So what is after that? Your memory cells. So whenever there is a reinfection with the SARS-CoV-2 virus, these memory cells again will produce the antibodies because they are recognizing the antibodies. So the role of the vaccination is to stimulate our body to stimulate the memorizing antibodies. So for your question, whether you should go for the second dose or no, ideally every CDC or any other WHO organization or FDA, they are not saying that you should not go a frequent booster doses. One booster dose is enough to get a vaccine protection against the infections.
Thank you so much, Dr. Satyam. So well explained. Thank you so much. And now, ladies and gentlemen, I will hand over to my esteemed colleague, Dr. Wilku, for more pertinent questions. Dr. Wilku.
Thank you, Professor. It was a wonderful session by both Dr. Hafiz and Dr. Satyam. Generally, it's a very hot topic about antibody testing and vaccination, especially the booster doses. Every day we are coming across a lot of questions, and we have received many questions in the mail. And first I will take from a chat. Got a call in between. Yeah. Mr. Harshad Singh is asking, I'm confused about antibody test results. After complete doses of vaccination, my SARS-CoV-2 antibody level was 125. Still, I got COVID positive. Was my antibody level not sufficient? I think Dr. Satyam and Dr. Hafiz both can put your opinion on this. The gentleman had antibody level of 125, still he got positive.
Dr. Wilko, as I just told earlier that your antibody titer, doesn't matter that what is your antibody titer. These vaccinations or by natural infection induce the antibodies usually does not prevent you from getting the infection. What is the meaning of these antibodies? That means if you are getting the infections, the severity of the disease will be very much low. So if if somebody is thinking that if I having 125, if I having 10,000, if I having 15,000 antibody titer, that means that doesn't mean that you will not get the infection. But yes, definitely, if you are having more antibody titer, that means they are more fighting against the infection. So that basically you have to understand. Second thing is that, as I just told, that this memory cells, these are memory cells, these are the factories. So when you are getting an infection, so that will generate the new antibodies. So if you are having right now 125 without the infection, and after a period of time when you're getting the infection, the antibodies titer will be pronouncedly increased. That will help to fight against the infections. So ultimately, they'll all depends on your individual immune status.
Okay. Yeah. Dr. Hafiz, your point, please.
Yeah, I think Dr. Satyam rightly put it. There's one more thing to add, just to add up, is the timing of infection. Sometimes people get infection soon after the vaccination, maybe in a matter of 2 weeks, and that time the body just takes time to produce the antibodies. So many times we get infected just in a matter of 2 weeks after the vaccination, when the body has not produced enough antibodies. And secondly, one more point which is to be considered is that maybe when we were infected, it was a Wuhan original virus, and then the virus mutated and you had a Delta or a UK variant, and you may have an infection with that different virus. So that's the only thing which scenarios which can be possible.
Yeah, thank you so much. This was quite obvious when vaccination started, and many people were complaining. I have taken vaccine, and immediately within a week I got positive. What my understanding was during that period, people when they got vaccination, they were free in a mind that they are not going to get it. So they were skipping all the precautionary measures, and that's what the reason they got infected. My understanding, people become negligent about this.
Absolutely.
Now, second question is, again, Mr. Harsel Singh is asking, is it necessary to have antibody test before going for a booster vaccine?
Dr. Viku, this is not required. Booster vaccinations, as I just told, is a specific people and at the end of the 6 months. So it doesn't affect much here if you're just going to do a the test for the antibody titer before going to the booster dose.
Okay. Again, there is one connected question because we received a lot of questions. I will take few of them because already in FAQs, Dr. Satyam, you have put all the answers. And Professor has also asked one question, which was very interesting question. Every day we face that. Okay. If fully vaccinated, how many booster doses are required? Because we are saying generally what MOH in UAE has informed people, if you have been vaccinated for both the doses, after 6 months you can go for a booster dose. Now we have come across many people, they have taken 2 booster doses. Initially sign a form of 2 doses, and after 6 months they took Pfizer, and again after 21 days or 28 days they have taken second dose of Pfizer. Was it required?
Dr. Vilku, this usually does not require to have the frequent booster doses. Okay, unless and until you are not working in a high risk population, like for example, if you are working in the hospital setup where you are contacting the disease very frequently, you are exposing to the patients of SARS-CoV-2 infections, and you want to make sure by yourself that my immunity is going down after 6 months, 7 months, and if I just getting booster doses, I will be protected. Routine population, booster dose single is enough.
Yeah. Yeah. Okay. So because even like you have already mentioned about the waning antibodies. So people, if those who are working in an area where there is a lot of chances of getting infected, so they should have some specific period of gap. Then they should go for the booster.
Yes.
Okay. And no, I would request both of you to put your option in this. Can I switch to 2 doses of another vaccine? how much gap? What I was asking, the same question has been asked. How much gap should I give for after the second dose of first vaccination? The like what you are saying, mix and match.
What the CDC is recommending that you should follow the same protocol as the primary vaccinations. Like for example, if you're going for Sinopharm and the Pfizer vaccination mix and match, that means it needs 6 months. For example, and if you're just going for Moderna and Pfizer, you are taking Pfizer booster dose after the 2 months of the Moderna. So what is there is a guideline is available on the CDC, if you can see that. So which primary vaccination we have taken it up, and if we are going to mix and match, so based on the primary vaccinations booster criteria, you should go for the booster doses.
Yeah. Dr. Hafiz, your opinion on this?
Yeah, I think as Dr. Satyam mentioned, there was also this was published in College Times, and if you've taken a Sinopharm and then you've taken a booster of Pfizer, a single dose, then you don't require another booster dose. It depends on which. So they're promoting a vaccine which is a DNA-based vaccine and a whole cell virus-based vaccine, so that the protection level is enhanced, and that's the science behind it. So if you're taking a Sinopharm, you can opt for a Pfizer or a Sputnik, which has a different mechanism of action, different epitope of the protein, and that's what the whole idea is.
Yeah, thank you, Dr. Hafiz. One more interesting question, Dr. Hafiz, you were mentioning about antibody spike testing and neutralizing testing. So generally, we suggest both the tests should be done in combination so that we understand better. So what is your opinion on that? How should we go for it? Should we go only for the antibody test, spike test, or we should do the neutralizing also?
The spike test alone, will not give an idea of the functional antibodies, because as spike is the most abundant, protein. Once you have the vaccine, it was being produced. But how much of that spike is actually functional, you will only get to know when you do the neutralizing. So as we do in our hospital, we try to do both the tests. So you may have, if someone is having underlying disease or immunocompromised or cancer on drugs, especially he may those drugs can inhibit his immunity, and these responses can be limited. So for those people, it's better to, especially not everyone. Does not apply to everyone. It's better to do both, because just having the knowledge of how much spike antibodies you have may not give you the functional information about how much of inhibition those spike antibodies are giving, how much of virus they can inhibit. That comes the 35% or 40% or 20% neutralizing inhibition comes from the neutralizing assay, and it's actually a reflex testing. So that's Dr. Satyam, maybe also he can add up something.
Yeah, so I'm agree with Dr. Hafiz that you should go for ideally both the testing, spike protein antibody testing and the neutralization antibody testing. But the one thing you should not forget here, that what time you have to go for testing. That means suppose you have taken a vaccinations after a week, if you are going for the vaccination, the antibody testing, no. Ideally, it should go 2 weeks after the second dose, or 2 weeks after the booster dose. And if you find that your antibodies are not developed, then you have to wait for minimum 1 month to see whether your antibody has been developed or not. And then you can just take the decision. You can consult with the doctor and you can get the advice.
Absolutely. Yeah, thank you, Doc. There is one similar question which I was asking just before this. One gentleman, Mr. Korean, is asking, he got 2 doses of Sinopharm. He got COVID after 3 months. Now, after 5 months of being positive, is it necessary to take a booster? If it is necessary, which one should I go for? Either Sinopharm or a Pfizer? It's a similar question, but only a little bit variant. Yeah.
If we can see in my presentation, I just told that what is the difference between the natural infection induced antibody and what is the difference between the vaccine induced antibodies. So if you just see, the natural infection induced antibodies are having the more widespread covering the antigenic part of the virus, because it is done by the whole viruses. So it means that if you are being infected 3 months after your vaccinations, You already developed the antibodies because of that virus. So no need to take the booster dose very soon. It will not help you much, because you should also not forget what is the content inside the vaccination. It is the same as
Yeah.
the inactivated virus or the part of the virus. We are not introducing any antibody inside the vaccinations. So do not expose your body very frequently to the vaccinations. You already been vaccinated 3 months before and now you are having the natural infection. So this natural infection induced antibodies are more strong, which is having both systemic immunity as well as the mucosal immunity, and it will last more than 6 months or 7 months. So you should not consider right now to go for the booster shots.
Thank you, Doc. There is one interesting question Mr. Sarswat Nasir is asking. He's more concerned about UAE. So once UAE authorities make the booster shot available for rest of the public, do you recommend that we take it?
Yes, we can do.
Okay.
There's no harm, yeah.
Okay, fine. I think that's clear. One more standard question every time we come across. One gentleman asked in a mail at support. So my grandfather is 73 years old and has a history of diabetes as well as hypertension. Is it safe for him to take a vaccine?
Yes, they can take the vaccinations.
Okay,
We
fine.
should not also forget that if he is contacting a natural infection, what will be the complications of the natural infection, and if we just getting the vaccination, and what will be the side effects of the vaccinations. So we have to see whether it is the side effects are much lesser than the complications of the natural infections.
Yeah. Thank you, Doc. We have received a lot of questions which are similar, which will be replying on the mail. If you have any other queries, so you can just mail us at support@rackhospitalrehab.com. We will definitely answer to you that. Thank you, both of you. Professor Kennedy, over to you.
Thank you so much, Dr. Wilku. That was a very enlightening question and answer session. Personally, I learned a lot from both the speaker sessions, and the added knowledge gained through the question answers was a bonus. I'd like to thank both our speakers. I'd like to add my thanks to Dr. Wilkus, to both our speakers, Dr. Hafiz and Dr. Satyam. I would also like to thank our audience of over 150 individuals who registered for this webinar, and this is our 16th webinar. I thank the speakers, I thank my audience. We could not have done this without our team, two of whom you can see on your screen. One is me and the other is Dr. Wilku, but there are several others who work behind the scenes, and I am deeply grateful to them. Now, if we could put up the post-COVID rehab slide. Do you have it? It was meant to be the last slide on this protocol. Well, in case we have not put it up, my apologies for that. And this is the online slide. I was looking at the physical rehab slide, but don't worry, I will explain this in a few words. The slide that you are watching is our online rehab program, and that is the webinars, basically. Now, we would like very much to support our audience with a physical rehab protocol, which is available again complimentary in RAC Hospital. This is part of our RAC Hospital Arise UNDRR offering to the community. So in case you would like to come in to us and take the support of our medical professionals, of our nutritionists, of our psychologists, of our physiotherapists, we would be happy to accommodate you in order to alleviate you or reduce your anxiety and symptoms relating to long COVID. That said, ladies and gentlemen, thank you so much for attending our 16th webinar, and we will see you again in approximately 2 weeks for our next webinar. Thank you and have a great day.
Thank you, Professor.
Professor, thank you, Dr. Bilku. Thanks, Satyam. Thanks a lot.
Thank you, Dr.
Good day.